as well, she was awarded the Teaching Excellence Award from IIT. And in 2018, she was appointed as the Dean of International Relationships at IIT Mumbai. And she's currently an invited member of research and training in WHO tropical diseases. So with that, she's here today to share her insights on nuclear import, a novel target in the drug discovery pipeline of Toxoplasma gondii, where I believe she'll be highlighting her research findings and expertise. So we look forward to your presentation. I warmly welcome you into the dais. And it's a great opportunity to learn from her expertise. Thank you.
You have the presentation here.
We start the presentation.
Directly.
I just, the opening slide, I can introduce myself.
You have the mic here. You have the clip here, you can zoom it in.
Right.
Forward slide, next slide.
And fine. Thank you so much. So can you all hear me? So I'm not going to stand behind the podium. I teach a lot. I'm from IIT Bombay, and our student strength is now 13,000. We have many, many departments, and we do a lot of teaching. We do a lot of research. We do a lot of admin. So I'm not used to standing behind the podium. I'm used to teaching. So I'll be walking around. So even though I told you about my institute, and here's a nice picture. This is just a few of our hostels. I have to tell you that I have really enjoyed my morning here with you at NIAB. So I met some of the faculty. Of course, I spent some wonderful time with your director. And I must thank her for inviting me, because I'll tell you where we met. We met at a panel about women in leadership. And I think we hit it off immediately. So we had such a good time. And I'm really grateful, very happy that she called me here. So I've had a really good time this morning. I've been meeting, people in the faculty. I want to meet the students as well. I saw your large animal facility, which was fabulous. The goats and the cows were terrific. And even though I come from an institute which appears to be many, many times bigger than yours, I want to assure you that we are all going through the same thing. So this is for the students. If you're getting frustrated because your reagents are not coming on time, same here. If the faculty are frustrated because the grants are not coming on time, same here. So we are all the same. We're all one. And I'm here to, really enjoy myself and have a great time with you today. I also want to say that despite all my responsibilities on my campus, the thing that makes me want to come to work every single day is the research, right? So I've always told my students that coming to the lab is like having a conversation with nature. So she's telling us her secrets, and we are slowly trying to understand her secrets through our experimentation. So for me, the thing which brings me the most joy, that's the thing which I've been doing for decades and that I love to do, is research. And so it gives me great pleasure to be invited here to tell you about what my lab's research is for the last few years. So the topic that I'm going to talk about is one of our lab projects, which is about nuclear import. We have some others. And again, it's great to be celebrating World Veterinary Day, because indeed this parasite that we work on, Toxoplasma gondii, it has a very broad host range, and it does infect domestic animals as well. So it's extremely relevant for what we're going to talk about. So these are two life cycles. This is the life cycle of the apicomplexan parasite of the Plasmodium species. This is the life cycle of Toxoplasma gondii. I love life cycles. I will not get into them in detail, because I'm sure we all know that the life cycle of a pathogen tells us a lot about the disease. So we'll not get into details. I'll only mention to you that this is obviously, this is the Plasmodium life cycle with the human in the center. But this is the Toxoplasma life cycle, and you can see all these domestic animals, as there's also cattle in this. Humans are certainly a part of this, but this cute little creature here, the cat, is the definitive host. So that's where the life cycle actually continues and carries out the sexual stages. So just to summarize, these are the two pathogens that I'll be telling you about. And the global prevalence is huge for both of these. So in fact, it is estimated that 1/3 of the human population is positive for Toxoplasma gondii. Toxoplasma does infect both human and domestic animals. It's definitely zoonotic. There are One Health implications for sure. And the other thing I will point out from these two is that these are complex life cycles and they have many different stages. So even within the human host or the domestic animal that we might be studying, there are multiple stages that, that are actually undergoing changes, right? So please keep that in mind. So I'm going to tell you a little more about these pathogens and the stages of their life cycles. And I'm going to focus on the clinically relevant stages. So this is malaria. This is the clinically relevant stage for the malaria, for the Plasmodium species. And this happens in the red blood cells. These cause disease. So that's when we go to the doctors with the fever. And there are also liver stages, which are there only in P. vivax as latent stages. And there's also these stages called gametocytes, which are needed for transmission to the mosquito vector. So the treatments are for the blood stages, clinically relevant, and these are the artemisinin-based combination therapies or ACTs. And it will not surprise you to know that there is drug resistance to all these drugs, not as widespread as for us to be scared yet, but there is drug resistance for all these drugs. The other parasite is Toxoplasma gondii. There are two clinically relevant stages I will tell you about among all those complicated life cycle stages. One is the tachyzoid. And under stress, this tachyzoid converts to a form called bradyzoids. These tachyzoids, they replicate very fast. These replicate slowly, the bradyzoids. And these are in a very heavily glycosylated cyst wall, so they form these latent cysts in the tissues. So in fact, bradyzoids can be ingested by us if we actually ingest meat which is raw. And this is one of the reasons why the incidence of Toxoplasma gondii, one of the reasons, the incidence of Toxoplasma positive people in France is over 90%, because they like cats and they eat raw meat. So tachyzoids and bradyzoids are the two forms of Toxoplasma gondii that I'll be telling you about today. So the treatment for Toxoplasma is a combination of sulfadiazine and pyrimethamine. And again, it should not surprise you that there's drug resistance. So having told you this background, I'm going to tell you a little bit more about the drugs that are used to treat these two parasites and why my lab works on trying to find new targets for drug discovery. So the current drugs and the cellular targets has been summarized in these two schematics. This is a little old, but it gives you the gist of the issue. So the ACTs, the artemisinin combination therapies, they target something called the food vacuole, which is in this Plasmodium, schematic here. And the other drug actually targets the folate biosynthesis pathway, which is in the cytoplasm. For Toxoplasma, both of the drugs target folic acid synthesis. And you'll see here that the drugs that are being used, they target either folates, protein synthesis, mitochondrial electron transport. Here we have some drugs also against mitochondria and other organelles. So if we are having to deal with drug resistance, we need to come up with new drugs in the pipeline so that we can constantly try to replenish the drugs which may fall by the wayside due to drug resistance. And again, we are all very comfortable with this because we are used to thinking about bacterial antimicrobial resistance, right? This is always a problem, XRD, TB, all that stuff. So we need new lead compounds so that we can replenish this drug discovery pipeline. And we want new compounds that are actually novel and do not target the same pathways again and again. So you can see on this slide, one of the pathways which is not being targeted is the nucleus, right? So these are eukaryotic pathogens. They have nuclei. And there is really no drug that is targeting this organelle, which is the nucleus. So I'm going to try to convince you today that nuclear transport is a novel target for drug discovery of these two apicomplexan parasites. Yeah, so I'll spend the next 30 minutes on that, maybe 35. So I hope by the end of the talk, I will have convinced you that indeed nuclear transport is a novel drug target, and I'll show you my data for that. Okay, so what is this nuclear transport? In eukaryotes, there's a transport cycle which has been shown in a very simple schematic here. It's much more complicated. There's a transport cycle which carries protein cargo inside and out of the nucleus. So this transport cycle involves this protein important alpha. It is a carrioferrin. And this binds to cargo. It also binds to another carrioferrin called important beta. And this heterotrimeric complex goes through the nuclear pore. And then there is release of cargo inside the nucleus. Now, important beta and important alpha, they are recycled back to the cytoplasm. There's a lot of details in this, which I have not given you, but this is enough for us today. So the cargo is actually recognized by important alpha through this sequence called a nuclear localization signal. And this is a stretch of basic amino acid residues, arginines and lysines. And this is the sequence that is actually recognized by important alpha, and it binds to the cargo, and therefore it is able to carry proteins, the cargo proteins into the nucleus. Okay, so let's remember important alpha, which you're going to hear a lot about, and the fact that they recognize cargo through nuclear localization signals that are basic residues. So nuclear transport is actually very important in many other disease, sort of systems. So I've just given a few examples here, but there is a lot of evidence for nuclear transport being dysregulated in certain types of cancers. And in fact, there are clinical trials in cancers for molecules that play roles in export of proteins. Yeah? So there are clinical trials already happening in cancer, in certain types of cancers. There's also links between nuclear transport and neurodegenerative diseases, in sepsis, in viral infections. So this is a pathway that has been targeted for other diseases. And obviously, our question is, can we target this pathway for our apicomplexan parasites that cause such widespread human disease, right? So again, I want to convince you by the end of the talk that indeed the answer to this question is yes. Okay, so let's talk about drugs for 2 minutes. So whenever we take any medicine, so how many of us have taken some antibiotic for a bacterial infection? Everyone, right? We've all done it. So when we take that antibiotic, we should realize, and we do realize, that they are actually what Paul Ehrlich in the last century, many, many decades ago, probably 100 years ago, he called these drugs magic bullets. And why are they magic bullets? They are magic bullets because they kill the pathogen in our body, but they don't affect our cells as much, right? So they have very little effect on human body, but they kill the pathogen. So they are magic bullets. They'll go and they'll just get the pathogen and leave everybody else safe. So for this to happen, drugs will actually bind to proteins in the pathogen. And then they inactivate those proteins, and therefore the pathogen gets killed. So they inhibit protein function. And if the protein is found only in the pathogen and not in the host, then the drug is a very nice magic bullet because it's able to quickly go and bind to the pathogen and inactivate it and kill the parasite. It will have no effect on host cells. And there are many examples of these kind of drugs for apicomplexan parasites. So I showed you this slide of the drug targets. This one. So many of these drug targets, you will find that they are only present in the pathogen and not in the human host. So here's the examples of these drug targets, which are found only in the pathogen and not in the human host. And these are artemisinin, which actually targets heme degradation, and also sulfadoxine, which targets dihydroteroate synthase, which is not present in the human body. Yeah, so we need to take folic acid from outside. It's because we don't have DHPS. So there are many drugs for apicomplexan parasites which have target only in the pathogen and not in the host. But this is not always possible, right? So there are other drugs which will have a protein target both in the pathogen and in the host cell. So you might think, now this is a gone case, this drug will not work because the target is in both the host and the pathogen. But it's not that difficult. It is difficult, but it is possible to overcome these difficulties because you will then try and design a drug which will inhibit the function of the pathogen protein, but not the host protein. So you need to bring in specificity, right? Your drug has to be able to only kill the pathogen, but leave the host cells untouched. So we need to find differences between the protein in the pathogen and the protein in the host. And there are examples of this for the apicomplexan parasites that I told you about. So actually, pyrimethamine targets dihydrofolate reductase, which is very much present in our bodies. And this is a drug which has been used for decades. It is also going to drug resistance right now in the last few decades, but pyrimethamine was used for such a long time to treat malaria. And it is actually a very remarkable story how a drug can very specifically target only the protein that is DHFR in the parasite and not affect our human DHFR. So it's a really fun story. I won't tell you now, but it's really fun to listen to sometime. So there are examples of both drugs which will only affect the pathogen protein, which is not found in the host, and drugs which affect pathogen protein, but then they leave the homologue of that protein in the host untouched. So we need to find differences. So let me tell you about importin alpha, because what is my talk about? It's about whether nuclear transport is a possible drug target for apicomplexan parasites. So importin alpha is essential in both pathogen and the host. It's a key nuclear transporter for cargo proteins. There's one gene in apicomplexans. It's expressed in these single cells. It's essential. There are 7 genes for humans, but they are also essential. So now I'm showing you a case where I'm trying to argue that this target, this protein target, is present in both the pathogen and the host. It is essential in both. So how can we use this as a possible drug target, right? So are there any differences? That's the crux. We have to find differences between importin alpha of the human and of the apicomplexan parasites. So I'm going to spend the next 10 minutes or 7 minutes showing you that there are indeed differences between these two proteins. Okay, so let's get into what does this importin alpha look like? What is its structure? And importin alpha proteins from yeast and higher eukaryotes, they have this very interesting property, which I'm going to tell you about. So this is a schematic of this importin alpha protein. It's got an N-terminal domain which binds to importin beta. It has a hinge. It has a C-terminal domain which is required for nuclear export. And it has this NLS binding domain. So these arm repeats, they bind the nuclear localization signals of the cargo proteins. So very interestingly, in this IBB domain, importin beta binding domain, there are also positively charged residues of lysines and arginines, and they resemble nuclear localization signals themselves. So because of this, you have a situation where you have NLS-like clusters, you have sequences which look like nuclear localization signals, and they will then go and bind to the NLS binding site in the protein itself. So what is the meaning of this? The protein has itself regulated its binding to its cargo, and this is called autoinhibition. Okay, so importin alpha proteins from yeast and higher eukaryotes, they show this property of not being able to bind to the cargo because of IBB domain, blocking cargo binding by binding to the NLS binding sites. Everybody clear so far? You all got it? There's autoinhibition, which does not allow cargo binding. So obviously your question in your mind is, how does cargo bind? So the way that cargo binds is, importin beta will bind to this IBB domain, and it will therefore relieve this autoinhibition and allow the cargo to now bind. So importin beta binding is a very important part of cargo binding. You have to have importin beta binding, otherwise cargo cannot bind. And this results in very nice regulation of nuclear transport. So I told you that importin alpha proteins from yeast and higher eukaryotes, they show this property called autoinhibition. So what about our apicomplexan parasites, importin alphas? So the IBB domains of importin alpha proteins, if you do a multiple sequence alignment of the proteins, you will see this is Toxoplasma gondii, Plasmodium falciparum, Saccharomyces cerevisiae, Homo sapiens, and Arabidopsis thaliana. These are the basic residues I told you about, arginines and lysines. So these are the 3 basic residue clusters in the IBB domain, which contribute to autoinhibition. You'll see that there are differences in the sequence through these different organisms, but let's focus on this cluster. You will see that in Toxoplasma, it is KKR. In Plasmodium, it is SKR. In humans, it is KRR. So it should strike you immediately that in Plasmodium falciparum, one of the basic residues has been replaced with a serine. This is the wild type protein. So there are differences. And what we did is we tried to understand what is the strength of autoinhibition of importin alphas from Toxoplasma gondii and Plasmodium falciparum, and are there any contributions of these basic residues. So I will explain to you about strength of autoinhibition for just 2 minutes. And I think you'll be able to pick it up very easily because it's quite an obvious concept. So how do we measure strength of autoinhibition? We take NLS containing proteins. This is all done in vitro. It's done in a test tube. We have purified NLS proteins. We have purified importins. But we have importins of two types. So we have the same importin alpha, which has got the IBB domain, the same importin alpha without the IBB domain. Okay, and then we look for a KD, an association constant of binding of NLS to this and this. So we get two different KDs. Everybody's with me so far? I'm just doing binding assays. Do you remember biochemistry from long ago? You've all done biochemistry, right, for the students? So this is a simple biochemistry binding assay. We are simply asking for the association or the dissociation constant of an NLS to a full-length importin alpha versus a truncated importin alpha without the IBB domain. If there is strong autoinhibition, the importin alpha having the IBB domain will not bind very well to the NLS. Agreed? And if it does not bind well, the KD is high. If you remove the importin beta binding domain, it will now bind very efficiently to NLS, the KD will be low. So if I simply do a division of the KD of the full length importin alpha divided by KD of importin alpha without the IBB domain, if there is strong autoinhibition, what do you expect the fold change to be? Very high KD for weak binding divided by very low KD for strong binding with delta IBB. What should that fold change number be? High or low? High, very good. You are exactly right. So if you have good autoinhibition, strong autoinhibition, if you do these binding assays, you will see a very big fold change if the protein has strong autoinhibition. If you don't have autoinhibition, what will you expect the fold change to be? Low. So we did quite a lot. We did a lot of studies on this, and they have been summarized in this one image. So these are our papers, but the summary of this image, this is like a thermometer of the fold change value of what I just told you. And Plasmodium falciparum importin alpha gives you a fold change value of 1. So it completely lacks autoinhibition, whether it is full-length protein or delta IBB protein, it binds with the same affinity to the NLS. So no autoinhibition at all. And we know that because if you make a mutant of the SKR to KRR and you do the same binding assays, you see 13 as the fold change value for autoinhibition. So autoinhibition has gone up with a single amino acid change. Now, Toxoplasma gondii has an autoinhibition value of about 3.45. And we think this is weak because if you make a mutant of this KKR to KRR, which is the same sequence as humans, it only goes up by a little bit like twofold like that. So it's actually weak autoinhibition. And the reason I call it weak is human importin alpha isoforms and even yeast importin alpha, they show this fold change value very high. So yeast will be 27. Right? Some of the plants will show you some 300. So the numbers are very high. Whereas over here, for our two apicomplexan parasites, this autoinhibition strength is either low or not at all. There is no autoinhibition. So what have I shown you so far? And, if you're interested, please read the papers. They are very, solid biochemistry papers. We've done a lot of SPR and other assays. So our hypothesis really is that apicomplexan importin alphas, they don't show autoinhibition. This IBB domain does not fold over and block the arm repeats. Whereas in host humans, there is strong autoinhibition, and this is actually the state of the importin alpha protein. So our hypothesis was that we can block with small molecules or peptides or whatever This X here represents a small molecule that can block the interaction between these two proteins. And this should be easier to do in a case where there is weak or lacking autoinhibition. It might be you need more of these small molecules to block cargo binding if you had strong autoinhibition. This was our hypothesis. So there is a difference between parasites and humans for this protein. Can we use this difference to find small molecules which block this interaction? So we started doing that. And I want to show you that what I will be telling you about is the proof of concept of our hypothesis. When I say proof of concept, I mean, I want to convince you that indeed I will be able to find small molecules or peptides that inhibit importin alpha and NLS interactions in a test tube. They inhibit nuclear transport in parasites in culture, and they also kill the parasites in culture. And at the same time, they don't affect human proteins and cells, right? That is the concept. That is our proof of concept, that they will only target parasites, but leave the human proteins and cells happy. So I'm going to show you data to support this proof of concept. So I just want to very clearly state that when I say proof of concept, I am very clear that these small molecules and peptides are not drugs. I'm not doing drug discovery. So drug discovery is best left to the pharmaceutical industry. They should do it. They are very good at it and they should do it. In the lab, we are not doing drug discovery. I'm simply showing you a proof of concept that importin alpha could be a good drug target for apicomplex in parasites. So whatever small molecules and peptides I show you, they may not be drugs. Some of them might be, but some of them may not be drugs. I'm simply showing you that this hypothesis is correct. I'm going to convince you that it should be correct. You may not believe me. Okay, so let's start with the first part of this hypothesis. First is I want small molecules that inhibit importin alpha NLS interactions in vitro. So for that, we worked with our collaborator, David Jans, who is at Monash University. And David has worked on importin alpha for his entire scientific career. In fact, David's lab was one of the labs which actually, in the pandemic, they screened small molecules for inhibitors of NLS importin interactions for SARS-CoV-2. So he is using this strategy to try to find antiviral small molecules. So David and his lab worked with Sujata, who was a PhD student in my lab. And she went off to Monash and she did what is called an alpha screen. So the bottom line of this is it's a high throughput screen. And we screened about 1,500 small molecules from different chemical libraries using alpha screen. And this was for inhibition of binding of NLS and importin alpha. We had importin alpha from Plasmodium, we had importin alpha from Toxo, and we had importin alpha from mouse. So we didn't have human importin alpha, David didn't have it, but we used mouse as the mammalian version. So after this screening, Sujata actually got quite a few small molecules that were able to block the interaction between importin alpha and NLS. This is summarized here. I don't want you to pay too much attention to this slide, but I just want to highlight a few small points. So the high throughput screen, when we checked the ability of different compounds to inhibit PF importin alpha binding to NLS, Toxoplasma importin alpha binding to NLS, and mouse importin alpha binding to NLS, we found small molecules that were able to inhibit all three at very, very low concentrations. We found small molecules that could actually block this interaction at a little bit higher concentrations like micromolar. And we found some compounds, especially Bay117085, that was very specific for only P. falciparum and not the others. We also tested these compounds in culture. So we now ask the question, can we kill parasites with these compounds? And the answer was very much yes. So in fact, some of these compounds were able to kill either P. falciparum or Toxoplasma or both. I want to highlight two things in this. One is that, and I will summarize this entire table on the next slide, but I want to highlight two things. One is, we also looked at the toxicity of these compounds on host cells. This is our human fibroblast cell lines. And we saw that if you divide the IC50 of host cells divided by the IC50 of your, parasite killing, you get something called a specificity index. So this just tells you that how much extra, how much extra drug do you have to put to kill the pathogen compared to the host. So your specificity index, if it is higher, it's better because you need a lot more of the drug to kill the parasite and a lot less of the drug to kill the host. So we got specificity indices which were not that great. Twofold, threefold is not great. You look at this, which is an actual drug for toxoplasma. This is pyrimethamine. The specificity index in our hands was 22, right? So we have some small molecules, but their specificity index is not that great. I wanted to keep it in mind because I'll show you something else towards the end of the talk, which is much better than this. And I also want you to keep in mind that these are definitely not drugs. Why? Look at the IC50 of dihydroartemisinin. It is 1 nanomolar. And in many assays, it goes down to 1 picomolar also, right? Whereas all our compounds are working in micromolar, low micromolar range. So they are not drugs. But they are small molecules which can block this interaction and they can also kill parasites in culture. Okay. So I've just summarized this entire table. So as expected, there were small molecules which work very well in vitro. They are able to block protein-protein interaction in vitro, but they did not work well in vivo, in the cultures. So what could be the explanation for this? There are cell permeability issues. They are not able to enter the cells, therefore they are not able to kill the parasite. So we are working with our colleague, Rohit Srivastava, on delivery techniques. We have small molecules which work well in vitro, and they work at even lower concentrations in the parasites. They kill parasites at very, very low concentration. So what could be the reason for this? There are multiple targets in vivo. In the parasite, importins are not the only target. These small molecules are going and attacking many different proteins. And finally, we have at least one compound which has got almost the same IC50 in vitro and in vivo, and we think this is something which may be actually an authentic target. So I've shown you the proof of concept. I've shown you that we found small molecules that inhibit protein-protein interaction and they kill parasites. They have some killing on human cells as well. So they are not great. They are not perfect. But they do definitely show these two properties. So now I'll show you, do they inhibit nuclear transport in culture? So very simply, the answer is yes. When we express, we make stable lines of these NLSs, these nuclear localization signals, fused to a fluorescent reporter like GFP. In both P. falciparum and in Toxoplasma, we are able to show that there is a decrease in nuclear transport for these two compounds and also for this Bay117085. This is not a very significant decrease, and I'm happy to talk to you later if you're interested as to why it may not be that significant. But there is definitely a decrease in nuclear transport in both of these parasites. So far, what have I shown you? That I have found small molecules that carry out all these three things. Okay. So now we tried a bonus experiment. We have these small molecules. We wanted to ask, do these small molecules inhibit other stages of the parasite? And if you remember the life cycles, they are very complex life cycles, right? So within the host, there will be multiple different stages of these parasites which are growing and differentiating. So do our small molecules target or kill not just one stage, but multiple stages? Now, why should this be even possible? So nuclear import inhibitors, they block cargo going inside the nucleus. Can you think, and especially the students, can you think of a few possible cargoes that might enter the nucleus to carry out their functions? What might proteins that have to go inside the nucleus? What kind of proteins might these be? Can you give a few names? Little bit louder, please. Ribonucleoproteins, I agree. Anything else? Some protein which has to go inside the nucleus to carry out its job. Transcription factors, thank you very much. That's on the board, on the slide. Are there any others that you can think of? Nucleases for doing what inside the nucleus? maybe chopping or tripping, DNA replication machinery, some things like that, right? Primases, whatever. So you guys are exactly right. There are many different proteins that have to enter the nucleus, one of which are transcription factors. And I showed you these different life cycle stages to make gametocytes and to become bradyzoites. The parasites have to induce the expression of specific genes, which happens through transcription factors. And these have been well studied. For gametocytes, it is AP2G transcription factors. And for this bradyzoid formation, it is this transcription factor called BFD1. So what has to take place? Important has to carry these transcription factors into the nucleus. So we asked whether our small molecules, which kill these stages, these red blood cell rapidly dividing stages, and they also kill tachyzoids, do they kill the gametocytes and do they kill bradyzoids, or do they at least block their development, right? And the rationale is that they are based on transcription factor activation, which has to enter the nucleus. So the answer indeed is yes. So this is work done by Mansi, and she actually showed that these two drugs, so this one CAPE, and also ivermectin and auranofin, they are able to reduce the gametocyte formation. She did this in collaboration with Asif at ICGB, Asif Mohammed. And we could also show in the lab that we could reduce to some extent, not completely block the development of bradyzoids. So you can see here, these are toxoplasma bradyzoids. They have been stained with DBA lectin because they have very heavily glycosylated cyst walls. So indeed, CAPE, BAY11, 7082, 7085, they do cause a reduction in the formation of these differentiated stages. And also, there are some hope, there's hope for compounds like ivermectin and auranofin, which also seem to block gametocyte formation, right? So we got a bonus over here. We could show that we are able to kill fast dividing stages of the parasites, but we could also block the differentiation of these latent stages and gametocytes using our small molecules. Okay, so summary so far, we have some small molecules. Do they inhibit interaction of important alpha NLS in vitro? Yes, many. Do they inhibit nuclear transport in culture? Yes, 3 of them. And do they kill parasites? Yes. And some of them in common over here also inhibit different stages of parasites. Now, the other thing that we said was they should not exhibit these properties for human cells. But I just showed you in my tables that the selectivity index was not so great for our small molecules, right? It was 2 and 3 and maybe 11 for BAY11, 7082, but we were not so excited. We're not that happy. So let me tell you another part of the story. And this part of the story, it comes from an observation that Mansi made when she was doing these experiments. So she was taking, with Asif and Shilpi, she was taking NLS fused to GFP on a plasmid, transfecting it and trying to make stable lines in Plasmodium falciparum. She was taking NLS fused to GFP on a plasmid and doing transient transfection in toxoplasma and trying to see whether she was seeing the nuclear localization, which she can now block. While she did these experiments, she found that when she used the SV40 T antigen NLS, and this is a nuclear localization signal which has been studied for decades in human and yeast cells. It's a classical NLS that everybody uses. When she took that NLS and tried to make stable lines, or she tried to do these transient transfections to see GFP positive parasites, she failed miserably. She never ever got parasites. She just didn't get them. So she was frustrated. So what we did was we actually changed the NLS over here to another NLS. And I'll tell you the trick that we did over here in the next few slides. So I want you to just remember this, that when we were doing our experiments, we were met with massive failure. It just didn't work. And we could have just said, oh, this is horrible. This is a flop show. We just want to give up. But we kept it in our mind. We remembered it and we decided to come back to it later. So I'll show you what happened. So what did we do? So Mansi, as she was doing these experiments, she realized that she was not able to make any stable line with SV40T antigen NLS. And she then decided to study this further. And she expressed two GFPs fused in frame with each other. This is transient transfections in toxoplasma. Each of these little banana-shaped things is one parasite. So these two GFP proteins, they were expressing very beautifully in toxoplasma and transient transfection. Now she took her SV40 NLS, T antigen NLS, fused it to these two GFPs, and she just saw these small pycnotic little dots which were GFP positive, which were co-localizing with the DAPI stain. So this construct never gave her viable parasites. They were always dead and unhealthy. In contrast, if she put the NLS in the middle of the two GFPs, she now saw parasites which were alive. And in fact, if you do a quantitation of the same data, I've just shown you one image, but I'll show you the population numbers. If you do a quantitation of percentage of GFP positive vacuoles, if you have just 2x GFP, you get about 20%. As soon as you put SV40T antigen NLS, you get nothing. She got 0 GFP positive parasites. In contrast, when she put the SV40 NLS in the middle of the two GFPs, she actually saw a little bit, she saw about 10% parasites, but they were healthy. There was still killing, but she at least got some healthy parasites. So in fact, these were the parasites that she used for those experiments on the small molecules. Because she at least saw something, and so she was able to carry out her small molecule experiments. So this was very, very surprising to us because we were using SV40T antigen NLS as an absolutely standard classical NLS that everybody else uses. And what did we discover? The same NLS is killing our parasites when we express it in the parasites. So we decided to look at this further. And what we did was Janice, who was a postdoc in the lab for about 6 months, she made these mutants of the SV40 NLS. So this is the sequence of the SV40T antigen NLS. It's just 7 amino acids. She made mutants that we knew from the literature would have less and less binding affinity for importin alpha. And they also had a lower and lower NLS score by bioinformatics software. So when these NLSs, these mutants were made and they were fused to GFP and transfected into parasites, we now saw that if you make mutants in the SV40 NLS which don't bind to importin alpha, you now start to see lots of GFP positive parasites, again 20 to 30%. So mutations of the SV40T antigen NLS do not result in toxicity. It's highly specific. And we wondered, what is this SV40 NLS, right? So is it that there's something special about it? Or is it that it's a very strong NLS and is therefore hijacking the import machinery? So Mansi did bioinformatics of the entire T. gondii nuclear proteome. This is a heat map of the strength of the predicted strength of the NLSs. And SV40T antigen NLS is pretty high. But there are many Toxoplasma gondii NLSs which are even higher in strength than SV40T antigen. So the parasite itself has many strong NLSs as well. But what she saw from her bioinformatics was these strong NLSs, they are all found in the middle of the protein. None of them was at the N terminus, right? So this is very similar to us putting the NLS in the middle of the two GFPs. It seemed to reduce the strength of the NLS, and we were able to actually make, express this in the parasites. So we really believe that there's something unique about the SV40 NLS. When we express it in Toxoplasma gondii, we kill the parasites. And this killing is happening within a few hours. It's within the first few hours of the life cycle. So there's something special about this SV40 NLS. So we then said, let's quickly figure out if this is going through the nuclear transport pathway. And one way of doing it was that we overexpressed importin alpha. So what is the hypothesis here? SV40 NLS, when you express it, it is binding to NLS, to the importin alpha of the parasites and preventing the nuclear transport from happening. So if we put more copies of importin alpha, can we rescue this toxicity? So that's the hypothesis. You have your NLS, which is binding to importin alpha and not allowing the parasite cargo to go in the nucleus. Now we overexpress, now the parasite cargo will go in the nucleus and the parasites will survive. So just to remind you, without overexpression, SV40 NLS with 2x GFP, 0 parasites, we couldn't see a single healthy parasite. When we overexpress importin alpha, either wild type or different versions of importin alpha, and this is a second copy, we now start to see parasitophorus vacuoles, which are GFP positive. I also want you to note that all of the GFP positive parasitophorus vacuoles are also positive for importin alpha, which we are checking here through immunofluorescence for the importin alpha extra copy. So you will see that wherever there are green parasites which have got SV40 NLS GFP, they are always co-localizing with higher amount of importin. Okay, so we were able to actually rescue our death, our toxicity of SV40 NLS T antigen by overexpressing importin alpha of different strengths of autoinhibition. And again, this population data will just sort of summarize to you that if you have only GFP, you will see only green cells. As soon as you put SV40 and SGFP, you will see no green cells, but you now start to see cells which have got both importin alpha and SV40 and SGFP because the importin alpha is rescued. And I want to end with this, which is, I've told you that SV40 kills toxoplasma very efficiently. In the literature, SV40 T antigen NLS has been used in human cells all the time. We wanted to confirm this ourselves. So what we did was, Mansi did this very crazy experiment. She did protein transfection. So what is the meaning of protein transfection? You are all very comfortable with DNA transfection. You take a plasmid, you introduce it into your favorite organism. So what we did was we had all those proteins sitting in our -80 freezer because we were doing biochemical assays of protein-protein interactions. So Mansi took SV40 NLS GFP, which was sitting in the freezer, and she used different concentrations of this protein. She simply incubated the host cells with this protein. And only thing she changed was she put 20 millimolar HEPs in that buffer. And because of the osmotic shock, perhaps, I don't know what it is, there is actually a intracellular uptake of this GFP inside the host cell, and you can see all this green, you can see the green signal inside the host cells, right? So this is the DAPI, this is the nucleus, and around it you'll see all this green signal. That is actually the extracellular proteins which were taken up by the cell when you put 20 millimolar HEPs. So we are going to do it with DNA transfection as well. But this was so easy to do because we had protein sitting in the freezer. So we said, let's try it, and it worked. And Mansi did a classical MTT assay, which is an assay for viability of host cells. And she actually saw that even up to 100 microgram per ml, 100% viability of host cells. So this is in the literature already, but we are also showing that the same NLS, which is killing Toxoplasma gondii within 2 hours, has no effect whatsoever on the host cell, nothing. Okay, so I'm done, very close to being done. So I want to just summarize quickly. I've already shown you this part. Now I'm going to just summarize the SV40T antigen NLS peptide. Over here, it kills parasites in culture really fast by targeting importin alpha. Why? Because if we overexpress importin alpha, we can rescue. We know that it does not kill human cells. And therefore, we know that the selectivity index is very high. It does not kill human cells at all at the concentrations which we have tried. So we are actually very excited about this SV40T antigen NLS peptide because it actually has all the properties of a quote unquote drug, right? It's highly specific for the parasite. It doesn't kill the host. It targets a pathway which is essential. And we've done a lot of things to show that. So this is the last slide. I think I made it in time. I hope our data has convinced you, right? Because I don't want you to just believe me because I said it. I hope the data has convinced you that indeed importin alpha is a drug target for apicomplexan parasites. Why am I saying that? I found small molecules, especially CAPE for P. falciparum, Bay117085, and these SV40T antigen nuclear localization signals for T. gondii. These are able to block this interaction. They are able to kill parasites. They're able to block nuclear transport, and they're able to also affect differentiated stages. So we have a large panel of properties of these small molecules and peptides. And what's next? For Bay compounds, we really want to try chemical modifications because they're very interesting. And if we can modify some of the groups with some, chemistry experts, we'll try and make them even better. We want to select certain small molecules and test them in animal models. And there are some molecules like ivermectin, moxidectin, or anafin, which are already in clinical use. So we will go back and check those panels and see if they're worth testing in animal models. And we really want to see if we can deliver SV40T antigen nuclear localization signal peptides. Just that 7 amino acid peptide, can we deliver it into parasites in some interesting way? So that will be a delivery system issue. And we're again working with Rohit Srivastava on that. So I'm going to ask all of you, have I convinced you that importin alpha is a good target for apicomplexan parasites? That's what I started with. I want to know if I've been able to convince you of that. You don't have to say yes or no, because science is never yes or no. But I hope I've convinced you, and I'm very happy to discuss it again, when we have a chance to meet in person, any of the details of what I've been saying. So again, that's the hope that I've been able to convince you of this hypothesis, which we started out with. And this is my last slide, and it is obviously the most important slide because all this work is done by all the fantastic PhD students who are in my lab. Most of this work is done by Mansi Bhambe, so she's just going to defend her thesis very soon. And she then done an enormous amount of this work, and she's really a terrific PhD student. A lot of the screening was done by Sujata in collaboration with David's lab. So she is an IITB Monash joint PhD student. We have a very nice program with them. Some of the work was also done by Vishakha. She is a student from Bangladesh. So she came on a TWAF scholarship to the lab. And Diksha's work I've not shown you. And also some of the work was done by Anupama. And I want to acknowledge the funding. So for this project, the majority of the funding has come from CERN and from ICGB Trieste, which is why we got that nice collaboration with Asif Mohammed. And we were able to travel, and Mansi could stay there for 2 months in their guest house and have a nice time. So thank you very much for your attention. And I really am enjoying being here. I'm going to end by saying, if there's any way that I can collaborate with you, I would be very happy to do so, because you can see we have some animal models and drug delivery and things like that. So any possible collaborations, more than happy to explore. So thanks very much.
Thank you. Open for questions.
Yeah.
May I ask?
Yes.
I'm audible. So my question is this, I mean, I'm little bit, still I am not convinced.
Very good. You should not be convinced.
So because I am having 2-3 points now. So the point number 1, I mean, this NLS target. So what I was expecting now, I was expecting the structural difference between the human and the pathogen. Now, of course, I mean, the parasite part is very convincing. Okay. But that human part, because, where it is actually,
Yes.
where that this molecule is binding. Now, second thing.
Can I just answer that quickly? So, Diksha, I didn't show you her data, but she's actually doing a lot of modeling. Her entire project is to do structural biology of
This NLS.
Of the NLS binding to importin alpha. She's also done modeling, and she does see differences between the human importin alpha binding to SV40 NLS and toxoplasma binding to SV40 NLS. There are differences, but I didn't show it because
that part is missing.
It's happening.
Now, second, the another part which is missing. So, okay, we are talking only about this NLS. NLS of this pathogen, NLS of this human. My question is this: if we will skip this human part, NLS of human, what is the percentage homology at least of this pathogen NLS with the entire proteome of human? Why I am asking? Why I am asking? Let me complete. Because Asif is doing this. Asif is very much involved, and he might be already knowing this. So why I'm asking? Because this small molecule is targeting NLS of pathogen. Okay. We are only focused.
It's targeting the interaction between the NLS and the important alpha. Some of
Of
them,
pathogen.
yes.
Huh.
So some of them may be binding to important alpha, the small molecules. Some may be binding to the NLS, but they are blocking the interaction.
This is this small molecule is blocking the interaction
Correct.
of cargo and NLS.
That's right.
That's right. Na? In pathogen. My point is this: this NLS protein is having any of the, I mean, is this NLS having some similarity with the entire proteome, any other protein of the human, of the host? See, we are focusing only.
Are you asking about the small molecule or the SV40 NLS? Which one?
NLS.
SV40 antigen NLS. Perfect. हाँ.
तो इसका अगर सिमिलैरिटी है जो NLS का जो इसका सिमिलैरिटी एंटायर प्रोटीन में अगर हम देखें.
Yeah. So let me answer that question. So, even though it's only 7 amino
Right
acids.
now, see, 7 amino acids, well conserved. But right now we are only focusing on this pathogen NLS and the NLS of human. That's it.
We are focusing on the SV40T antigen NLS.
Of pathogen.
Oh, it's not SV40T antigen NLS of pathogen. SV40T antigen NLS comes from simian virus 40, which is an adenovirus. It's a completely different NLS. It doesn't come from either of these two organisms. Right. And in fact, if you think about it, the human cells, they get infection by adenovirus, and that SV40T antigen NLS is being used in the human cells to deliver viral proteins to the nucleus. So it is like the virus is using its NLS to deliver to the human.
Ultimately, the target is NLS.
It's not NLS. SV40T antigen NLS is a very specific sequence. And it is the only sequence so far that we have been able to kill parasites with. We have tried a few others, they don't kill the parasites. There's something very unique about SV40T antigen NLS sequence, which is killing Toxoplasma parasites very efficiently.
So what is the similarity of this protein in the entire proteome of human? This is my question.
No. So again,
this is you checked or not?
We have not checked human. We have definitely checked in Toxoplasma. It is not at all there in Toxoplasma.
That's why, madam, it is specific for the Toxoplasma.
No. So again, when you say what is the similarity in humans, see, so we have not checked all of the analysis of hosts. So I just want you to remember that SV40 virus infects human cells. So that SV40 NLS is inside human cells in an infection a lot. And the virus NLS does not kill human cells. Yeah. I mean, we have not checked the entire human proteome. We can talk about this later, I'm sure. But, it's an NLS which the human cells see a lot when they get viral infection. SV40T antigen viral infection.
Thank you.
Yeah, let's take some student questions.
The SV40T analysis is taking in binding to the importance in the toxoplasma, and they are not letting the basic proteins which the toxoplasma requires to avail the importance. And if you supply importance from outside, it's just risking the effect.
Copies. We give more importance.
So what? So don't you think that in the long run, if it goes as a drug, the toxoplasma itself will start producing more importance?
Question. What an excellent question. Thank you. So we have thought about that. I didn't show you this data, but this data, which was of the rescue, this data, which is rescue data, we can only rescue in transient transfections. And in transient transfection, we have also done image analysis of the intensity of the expression of important alpha. And in transient transfections, it's much higher. It's almost 10-fold higher. we have the same experiment done with stable lines where we are expressing these important alphas on the chromosome. And in those stable lines, the cells cannot, I don't think they can tolerate so much important alpha. So they have down-regulated to some level. And when we do a rescue experiment in stable lines, we cannot rescue. So that's a really terrific question because the parasite will try and upregulate important alpha and overcome the toxicity. But it appears that when you make stable lines, the parasite is not able to handle as much important alpha as it can when it does transient, when we do transient transfections. So this is a question for drug resistance. And it's a great question because today, when anybody talks about drug discovery, they bring in drug resistance in the early stages. So anyone who has a small molecule that they think is a drug, they start doing resistance assays right now, just to show that there's no chance of resistance. So we did think about it, and we are not able to rescue when we make stable lines because they downregulate expression.
Hi, ma'am.
Hi.
I have observed two things that was curious for me. One thing is starting when the IBP domain of either Plasmodium or Toxoplasma, when it changed to human one, the autoinhibition is not recovered as
That's correct.
compared to the level of human itself.
That is correct.
Is there any reason or are there other domain other than IBP domain which can contribute to this autoinhibition?
That's very correct. You have noticed the correct thing. And the answer is, you have given the answer yourself. The sequence of the two important alphas is very different. Even though they have the basic structure, it's not very different. They have a very similar sequence with arm domains, everything. But there are differences between the two organisms, definitely. So this certainly says that simply changing one amino acid does not get you back up to the high level of human and yeast. So there are other amino acids for sure that play roles. And there have been many studies which show that the flanking sequences around these repeats, these basic residues, even the flanking sequences are very important. Right. So you can see here that there are many differences over here. So they are all contributing to that high level of autonomy.
Yeah.
But for Plasmodium, it's interesting that simply one serine residue can jack it back up again.
Yeah. The second thing is, do you have an idea about what is happening to the NLS when it is put in the middle of the protein? Why it is killing when it is alone or in the entire middle, but not if it is in the middle?
So, we only have an idea. We have not been, I don't know how to test this experimentally, but the idea is that the NLS, when it is placed on the N terminus, it is accessible to the transport machinery to important alpha. So it's just there, it's just available, it's just, it's very much accessible. Whereas when it's inside, it could be sort of embedded somewhere. It may not be, so actually GFP is basically, it's two, beta barrels. So it can't go in the GFP. But we think it is something to do with the accessibility of the NLS. We can't, if we do bioinformatics, we'll get the same score. Because the bioinformatics only looks at the NLS itself. It doesn't look at it in the structure of the whole protein. So we think it is an accessibility issue.
Thank you, ma'am.
Thanks.
Excellent talk, Swati. Here. So I have few questions. Like IBD, you said there is autoinhibition in the human, but no autoinhibition in case of Plasmodium and Toxoplasma.
Yes.
Although the domain is there.
Yes.
So what might be the reason for that?
So, another great question. What we really are doing now, which we are extremely excited about, but I didn't tell you, we are trying to understand what is the biological significance of lack of autoinhibition and weak autoinhibition for the parasite. Why is the parasite having an important alpha which is not showing strong autoinhibition? And we've done some preliminary experiments which are suggesting that if we put a strong autoinhibition, so if we have parasites where we express a second copy of different types of important alphas, I'm going the wrong way, sorry. So if we have parasites, stable lines of parasites which express different copies of important alpha. So we have parasites which have got wild type. This is lacking autoinhibition and this is stronger autoinhibition. 6, the fold change is 6. So we have stable lines where we have got 2 copies. One is the endogenous copy and one is this extra copy. Then what we do is we use CRISPR to knock off the endogenous copy and we see are they growing or not. And we've done this twice. And we have to do PCR, a lot of screening to make sure that it is absolutely correct. But we've done it twice, and the stronger autoinhibition lines, they just don't grow. So they're actually not growing if they have only a single copy of strong autoinhibition. So that's all I have now, but we're very interested in this question because why should these parasites have weak autoinhibition important alpha? And we think it helps them in their replication. If you look, we've also done some bioinformatics looking at NLS sequences, and we see some correlation is not great, but the NLS sequences, the strengths of the NLS, they vary in these parasites. They might be on an average, I think a little bit higher or something like that. So we are trying to answer that question. We're very interested in that question.
The second question is like between, as you said, important B is different. Some difference is there between Plasmodium and Toxoplasma, and when you have screened for the inhibitors or the compounds.
Yes.
For example, Bay is showing activity in Plasmodium. It is not showing any activity in Toxoplasma. The other one is CAPE is showing, I think, activity.
Yeah, they have some, yeah.
And then have you checked this SV40 tagged NLS in Plasmodium?
Yeah, so in Plasmodium, the only data we have is that we cannot make any stable lines. So if we have to repeat these experiments, we'll again have to go back with to Asif because he's the one who helps us to do these genetic manipulations more efficiently. So we haven't done that yet, but I will tell you what we want to do very quickly. So I'll just show you what we want to do. I'll just explain it very briefly. So this experiment where we were able to put SV40 NLS from outside to protein transfection, crazy experiment in HFF. What we are doing next, what Mansi is doing right now is she's trying to take these lines, these cultures, and she's going to infect them with toxoplasma. So she wants to see whether any cells which have got all this green signal of SV40 NLS GFP, do they not support parasite growth, right? So we were able to kill parasites by doing DNA transfection. That is not a therapeutic strategy at all. So we have cells now expressing SV40T antigen, host cells. We'll try and see if the parasite is not able to grow in such host cells. If it works, we'll do that in Plasmodium as well. Yeah, so it's in the works, but still early days.
Thank you. Thank you.
Yeah, thanks. Yeah.
Is actually working. So could it be possible that this SV40T antigen NLS binding to importin alpha in parasite? Because you said in the earlier that they actually exhibit weaker autoinhibition. Maybe having this NLS binding, maybe it is making it stronger autoinhibition on the importin alpha. So when you try to overexpress importin alpha rescue, you saw the rescue, right? So could it be possible that this binding has something to do in changing the weaker autoinhibitory form of importin alpha to stronger so that it is unable to, I mean, it may have the implications on that.
So, I've never thought of autoinhibition strength coming from an NLS. So we always think of autoinhibition strength as an intrinsic property of that important alpha itself.
Okay.
So I have not thought about this at all. I'm not sure if it's right, but it's an interesting idea, which I'm happy to think about when I go back to the lab. But according to me, autoinhibition strength is an intrinsic property of the important alpha itself.
Yeah, I'm just curious to know, like you mentioned in the earlier slides that you have designed certain in vitro experiments to understand the autoinhibitory strength of autoinhibition.
Yes,
so I was
we did
thinking
that.
if,
No.
So
the autoinhibition strength that we do with our biochemical assays, those change only with mutations of important alpha, nothing else.
Okay.
So those are all binding to NLSs, SV40, whatever.
Yeah.
And the autoinhibition strength is a property of the important alpha itself, for sure.
Okay.
But, different ideas are always fun to
Yeah.
think about. So I will think about that.
Another question I have, second question is about in continuation with Dr. Paresh Sharma's question, like what is contributed to the weaker autoinhibition in parasites compared to human. Do you think there might be some cellular machinery or secretory proteins in the parasites that are making this weak inhibition, which is not absent in the humans?
Yeah, so actually, that's another very nice question. I don't think there's any secretory machinery, but there are very nice papers showing that, in that first slide, I showed you that nuclear import and export cycle. What I did not say on that slide is that the cargo release is driven by a gradient of RAN GTP and RAN GDP in the nucleus and the cytosol. And there's this nice paper showing that this gradient does not seem to exist in toxoplasma. So there are other differences in these parasites for sure, which are different between, in that nuclear transport, which is apart from important alpha. So why weak or lacking inhibition is going to be a tough question to answer because it's the full biology of the parasite. So the RAN gradient seems to not be there. So yeah, there could be many things that the parasites are doing.
Hi, Shwati. Nice presentation. I think the question that I was cooking, they're already served.
Yeah.
So I got another question for you.
Great.
So with especially with toxoplasma, a lot of people they check this blood-brain barrier crossing of the drugs or peptides that you
Yes.
discover. So in your case, especially this SB40 NLS
Yes.
peptide, what is its what literature says in terms of its capacity to cross blood-brain barrier?
Yeah, I don't know. I have to be very honest with you. I don't know. I don't think it's a positively charged peptide. I don't know if it'll cross the blood-brain barrier. So the simple answer is I don't know. But having said that, actually NLSs, there are some great examples. I removed the slide, but there's examples of NLSs themselves being cell-permeable peptides. So there are, so HIV TAT is a classic example of a cell-penetrating peptide, which is an NLS. And it's being used as a carrier for carrying other molecules inside cells. So I didn't show this data, but we have tested that cell-permeable peptide, HIV TAT NLS, to see if it kills toxo in a transfection. And it doesn't. So if it had killed toxo, and it was also very high in its strength, it was around 10. So we were hoping it would kill toxo, then we could have made that peptide and it would have gone on its own from outside without putting HEPs and all this stuff. So to answer your question, I don't know, but I'm not very optimistic that SV40T NLS can cross the blood-brain barrier on its own. It can't even enter cells on its own. So I think that if this is at all interesting in the future, there has to be a tie-up for delivery. Delivery systems, I think, would be very crucial. And, I really feel that it would be so nice if we could have, platforms, right? So somebody is looking at some sort of lead compound, small molecules, peptides. Other people are looking at delivery platforms. It would be so nice if we could talk to each other and, I have something interesting, but I wanted to cross blood-brain barrier. I have something interesting. I wanted to cross the bradyzoid cyst wall. So one of the reasons why our small molecules may not have done as well as they could have is that they have to penetrate the cyst wall. Okay. So I would be very, it'd be very interesting. I'm not an expert, but I would love to have, discussions with people who work on delivery platforms.
Thank you.
Thank you so much. It's been a real pleasure being here.
May I now request our director, madam, to felicitate the guest, please.
Thank you so much. Thanks, everyone.
Before we take the group picture, maybe a vote of thanks. Thank you, Dr. Pankaj.
So good afternoon, everyone. It's my privilege and honor to propose vote of thanks on this special occasion of World Veterinary Day 2025. First and foremost, I extend my heartfelt gratitude to our director, NIAB, for conceptualizing and organizing such a wonderful lecture. And it's really like we enjoyed, as you said, that it will be a wonderful lecture to hear, and we really enjoyed it. And that shows that enthusiasm and the kind of questions that you have got. So everyone was very much attentive. So yes, and a special thanks to you for coming and delivering such a wonderful lecture. So thank you very much. We are also deeply thankful to the scientific community, students and faculty members for showing lots of enthusiasm for the lecture and joining over here. Specifically to the students for your curiosity, attentiveness, and the presence. Your involvement today reflects the promising future of the veterinary science. If you are if you remain focused for such kind of research. Finally, I would also like to acknowledge the contributions of scientific management team, this event management team, and the ICT administration and other staff. Because of their help and constant help and support, we have seamlessly organized this event. So thank you very much to everyone who have joined in person or virtually, and thank you very much. And please come forward for a group photo. Thank you very much. The background. Background slide. It's okay. Aram se kar. Yes.
I think Madhuri's voice doesn't reach to that corner to.
Please, students, please come. Students, please come forward.